Jun. You from So our next talk is by Jun Ding from Ziv Bar-Joseph's lab at CMU, and he'll be talking about reconstructing differentiation networks.
This one.
Okay. Yeah. Oh, your name.
Okay, this one. Okay. Thanks, organizers, for giving me to talk about the work. This is the generous paper published early this year. Is about reconstructing differential network and their regulation using single-cell, time series single-cell RNA-seq data. So in the very beginning, people actually are studying the cell lineage and differentiation using the microscope. So actually here you can see is one of the very first cell lineage in the C. elegans. And this work has been awarded in 2002 Nobel Prize. And they have chosen the C. elegans for a few apparent reasons. The first C. elegans is quite simple, only around 1,000 cells. And then what's more important, the C. elegans is transparent, so make it much easier to trace lineage using the microscope. But this type of method cannot be applied to a relatively complex organism, such as human or mouse, in which we have training those cells. So we don't have a very good strategy to this until we have this time series single-cell RNA-seq data. Here is an early paper published in Science this year. It's about reconstructing the cell lineage and the differentiation in zebrafish. So the question here is how we start from the time series single-cell RNA-seq data to get the trajectory, the differentiation network here. And not all methods have been developed on this. The most famous one are the monocole or the scuba, slicer, etc. All these methods actually are pretty good. They can give you very solid trajectory inference, but they have some teeny tiny limitations, as I mentioned here. The first would be this method, although they can give you the trajectory, maybe it's pretty solid trajectory, but it does not tell you what driving the cells to different cell fate. Why some cells go in this direction, but other cells go in the other direction? So this problem is very critical for some maybe biologists doing the cell reprogramming. And another issue, which I think is more critical, is all of these methods actually are based on owning the expression data. But we all know for the single-cell RNA-seq data is very, very super noisy. So we should not trust only on the expression data. So, and our solution here is we want to using an extra piece of information. And here is we want to using the TF regulatory information. To be more specific, is TF binding situation. So I will just illustrate using a very naive example to show why this information is helpful to mitigate the impact of the noisy. So suppose you have a time series data in 3 days, day 1, day 2, and day 3. You observe the expression for gene A at day 1, day 3. Actually, it's 10. But at day 2, you observe the 0 expression. Then you might start to wonder whether this 0 expression is a dropout, which is very common in single-cell RNA-seq data. But it could be the real expression if there's some significant regulating event happening at day 2. It could be possible this 0 is a real expression. So without actual information, we don't know this. We don't know the answer to this question. But what if we know the TF binding in the 3 days? Like the binding, the similar TF binding, this gene A and the expression for this TF are also pretty much similar. So then we should observe similar expression profile, but we do not. We observe a 0 here. That is very likely a drop of noise. So I mean, this is just a simple kind of idea to show this kind of why this regulated information might help. To integrate this, so based on this intuition, we developed our method called SCDiff. Apparently, I'm not good at choosing names, excluding my daughter's name, which I think is good. So SCDiff actually was composed by two major components. The first would be building up the initial model. And this part actually exactly the same as all others. We just, the initial, building up the initial model is also 100% rely on the expression data. But what makes the method unique is we integrate the TF binding information into the second part, which is iteratively refine the model using the TF information. And so just talk about the first part, how we build up the initial model, because this is not the unique part of the method, so I just need to briefly talk about it. So the first will be, just like all other methods, we start from the clustering the cells. But to clustering the cells, we need to know how many number of clusters in the data. So we don't have this prior-knowledge in most cases. So what we can do, how we estimate the K here, and what we're using our ensemble strategy. We just sample the data, like for just randomly choosing genes or even cells from the original data multiple times, say for 100 times. For each one, we're using some clustering evaluation metrics. Here, actually, we're using 3 here. Using these 3, we will choose the best K for each of these Ks. And for all the sampling, we will choose the best K. And using the K, we can do the clustering, get the clusters. But that's not done yet, because there's another issue of the single cell is always unsynchronized. Although the cells was measured the same day, but it doesn't mean the cell actually at the same differentiation stage. So to do this, actually, we're using a strategies called DTA, distance to the ancestor. So to do this, we might need some prior knowledge. We need to find a list of the root cells we think is the ancestor for all descendant cells. So we can use some marker genes to pre-select the root cells. But if you don't have this this kind of prior knowledge, these markers, don't panic. We can using some other like visualization, some diffusion map. You can choose the cell at the very beginning. Or if the first time point is far away from others, we can using all the cells in the first time as root cells. Anyway, once we select the root cells, we can calculate the distance from all other cells to the root cells. And the distance can be Euclidean distance, can be correlation distance, can be even diffusion map distance. And so although performance might be slightly different, but the idea would be essentially the same. So we calculate the DTA for all the cells in the cluster. Then we sort the clusters based on their average DTA, like this. And then for each cluster, we will have vector, because for each cell, we have DTA. And then we do some compare every neighbors to see whether they're significantly similar using rank sum test. If they are very similar, we think it's the same stage. Or if not, I think it's at different differential stages. So in this way, we can kind of synchronize the cells and the clusters. And once we synchronize the cells and clusters, we just connect. We're building a tree by connecting the clusters in the closest one in the preceding network, and keep doing this until we reach the root. So that would be our first initial model. So and then we just talk about the next part, how we iteratively refine the model. So we're using pre-select. We don't using all TF because for some TF we don't have enough information from the data. We don't the target for the TF. So we're only choosing the TF whose target gene are significantly overlapping with the differential gene in the initial model. So once we pre-select a list of TF as the candidate, we build up our multi-class natural regression model. So it's kind of classifier. So input would be the TF binding for each gene, and also the expression of this TF. The output would be categories, whether the expression should going up or should going down or not changing. We don't want to predict the exact value, how much going up, because it's too complicated, especially too complicated task, especially in a single cell noisy data. So once we get this model, we just plug it into a model called Karma filter. So the idea of this model actually is coming from the GPS, the navigation system. So actually, the GPS the data actually is kind of similar to the single cell RNA-seq. So the GPS keep pulling the signal from the satellite, and the satellite signal actually is very noisy because of the long travel and the impact of the surrounding environment. So that data are always very noisy. But what GPS does is does not 100% trust on the signal, satellite signal. They're always using the direction of the previous position and the direction of the car and also the speed of the car to estimate the potential, or current position. So it has an extra piece of information to estimate the current position. To similarly, we have the same. We don't trust 100% of the direct single cell measurement. We kind of somehow using the extra information, just like the speed and the direction of the car. We want to use the TF information to somehow to predict the current expression based on the expression and the parent state. And also we have that means we have two pieces of the evidence. One evidence is from direct observation, the measurement, and also we have another estimation of the current expression, the expression of the current state. State means the cluster at the initial model. So that means we can estimate so using the regression model, we know where the expression of the current state should be similar to the parent state or should be larger, should be smaller expression than the parent state. Now that's one estimation. And also we have direct observation. So the common filter is kind of summarized. It's kind of weighted sum. You just choose, should we trust more on the direct observation? Should we trust more on the estimation from the parent? And also we're doing forward and backward karma smoother. So that means we're not only using the parent information, we are also using the descendant information. So, and that's pretty much the way how we re-estimate expression for each cluster, not only based on the expression. Here, actually, before we apply this to the real data, we simulated on very noisy data, and we applied it. It seems very good, and the estimation is very close to the truth. And so once we re-estimate the expression for all these kind of states, the cluster here, we will reassign the cells back because the initial cell assignment is based on only the expression data, the clustering, right, coming from the clustering. Then here, we just reassign because we re-estimate expression for each state. That means we can reassign the cells back to the state. So some cells will get reassigned. So that means the assignment of this model, kind of cells in the cluster will be different. Then we need to re-estimate the expression of these clusters, the states. So that means kind of iteration. We keep doing this. We keep doing re-estimate and reassign until it converges. And that will be our final model. But in this process, actually, the regression is also working, the classifier. So lots of regression. So the coefficient for some TF may be shrunk to 0 if this TF is not important. So in this process, it kind of will select the most important TFs. It's for the kind of differentiation process. So that pretty much is the model. Then we apply the model actually to a bunch of data set. Here we only talk about the non-data set. The reason why, because our collaborators actually is working mostly on the long development, so it's easier for them to design experiment to test our prediction. So, but we do have the result for other data set, a bunch of data set in our original student research paper. So we predicted all of that. Everybody knows this. We predicted ciliated and clara cells actually differentiate much earlier than 81, 82 cells. And we predicted a list of the no regulators for the non-epithelial differentiation, like E2F4, NKX21, etc. So those are just to know everybody knows this. So it's far away from convincing. So and to make it more convincing, so we just do some standing result. A standing experiment. So here actually is our HIF1A, the expression of the HIF1A predicted in our model. Here I see it's significantly lower in the E18.5 compared with the earlier time point, E16.5. So, and then we started thinking, and also, not only that, we also predicted this as an important regulator in our model to regulate the differentiation from E16.5 to E18.5. Then we started wondering why is we are significantly lower? What if we overexpress this TF, what will happen? So we overexpress this TF in this time from 16.5 to 8.5, and we observe here the green is the marker for 81 cell, and the red is the marker for 82 cell. We do find the distribution of this 82, 81 actually is the non-epithelial, two different non-epithelial cell types. So we do find this kind of disruption of the distribution of these non-81, 82 cells. But not only that, we find the survival of the mice significantly decreased as well. So that's kind of validated. If we lock out this TF, HIF1A, at the time we predicted, it will have significant impact on the non-differentiation, just as the model predicted. So that's just the one experiment. We also do some log code experiment. We log code this TF and see whether we will have big impact. So this TF we predicted have impact on the differentiation of the 81, 82, and also Clara cells, but not for CD80 cells. And those just non-epithelial cell types. So and we here actually those are 81, early 81, and this is 82, and this is the 81, and this is the CD80, this is the Clara. So, but we don't have a very big significance. Here, larger means larger impact on the result. But for this one, it's not that significant. If you look at this one, HMGA2, almost no impact on this one, this path. So very significant. So result here. So that's kind of direct evidence to our predictions. But we also have other piece of evidence is the go terms. We perform the go terms for the all kind of predicted TF. We perform the go terms for the predicted TF and find it's significantly enriched in this categories, 3 categories. Actually, the regulation of the epithelial proliferation and cell proliferation, and also differentiation. You see here because of the color, I think. So it's all related to the lung development here. And also, we have another piece of evidence. We have the NKX21, which is known to regulate in the lung differentiation. We have the activating target based on experiment, chip experiment. So it's really activating those targets. And we compare those activating target in the NKX21 in the lung development. We find is a significant overlapping with the differential gene in these 2 passes. And these 2 passes was already was also predicted by our model to be regulated by the NKX21, exactly the same. Exactly the same actually. And so what's more importantly, I will just put it at last, our model is very robust. If you still simulate the insane dropout, 80% dropout, we still have 16% of the agreement. That means you still have almost 60% similarity with the original data. And the Gaussian noise will be even better. So that means pretty robust result. The model is pretty robust. So to summarize, so our method SCDF actually using the actual TF information because using not only trust 100% on the expression, so it's more robust to the noisy. And the method SCDF is able to predict the regulating TF and also is most importantly is the regulation activation time, which is very critical to the cell determination. And this tool is available here, and this is our paper. And lastly, I thank my advisor, Dr. Joe, mentor Zil Barjoseph for the mentoring, and for our thanks to our collaborators, Naftali, Jeff, and Bruce for the feedback of the model and the validation. And also thank Michael and all the team, other members in the team for the valuable suggestions. And thanks for the founder of NIH and the ISMB for the travel ship. So, yeah.
So I was just curious if you took all your samples instead of, so what you're doing is clustering per time point. So if you were just removing the time label and glomming all your cells together and then applying something like, t-SNE, or do you see like the major populations, the time actually being the major,
yeah, that's a very good question. Actually, we have some collaborators, they don't have time series, they don't have one.
But for your data set, I'm just wondering if there are like cells that are, if this structure that you're imposing by clustering per time point first and then trying to relate them is too stringent, or whether maybe the times are so separate that the transcriptomes are very different.
So what's your question is whether this time is an important thing, right? That's your question?
Well, I was just trying to see if you took all your cells together and clustered it, whether the populations that would come out would group by time.
That's a very complex situation.
I'm just curious if you.
Yeah, it's not always the case. Some data set, yeah, the clustering always very good match with the time and the measure. But the cells, the clusters was just realigned to the previous time. That's happened a lot. So it depends on the experiment, how the time spent between you design the time series experiment, right?